Hi all, my name is Karen Gonzalez. I'm currently a senior majoring in biology and minoring in chemistry at Catawba College in Salisbury, North Carolina. Today I'll be talking to you a little bit about a compound called azelaic acid and its potential antimicrobial activity on the escape pathogen panel. Before diving into the findings, let's establish a basic understanding of AZA as well as what we mean by escape pathogen panel. Azelaic acid is a saturated dicarboxylic acid, typically found as a white powder, and it also occurs naturally in grains like wheat, rye, and barley. Its structure limits skin penetration, hindering its effectiveness when used alone. However, once it permeates the skin, studies show that it has antimicrobial and anti-inflammatory effects. Notably, it has not yet been tested against the highly virulent antibiotic-resistant pathogens known as the escape pathogens, which include Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacter species. We'll begin by talking a little bit about why exploring effective and safe antimicrobial compounds is crucial not only for society at large, but also for advancements in biomedical sciences. Then, using a couple of figures from past literature, we'll take some time to dive deep into AZA's mechanism of action in order to learn more about its properties and applications before talking about my research. Lastly, I will share with you my hypothesis, methods, and results, and discuss what kind of things I plan to do moving forward. As we know, the skin, our body's largest organ, plays a vital role in overall health, so keeping it healthy is in everyone's best interest. However, common skin conditions like acne are among the top reasons patients seek primary care, as noted by physician assistant Mrs. Echeverria in the Journal of American Academy of PAs. Furthermore, managing conditions like acne is essential not only to reduce physical symptoms, but also to prevent the mental distress associated with post acne scarring, which can impact quality of life and social interactions. Skin conditions are common in primary care, as we established earlier, and factors like diet, environment, or hormones can all disrupt skin flora, making it challenging to determine the root cause and find effective treatments. Additionally, there's an increasing resistance to conventional antibiotics, highlighting the need for novel compounds. This is where azelaic acid comes in. Let's explore the mechanism of action of AZA and how it differs from traditional antibiotics, which usually target bacterial proteins or cell wall structures. AZA, in contrast, operates through multiple pathways, as explained in past literature. First off, it interferes with bacterial DNA and RNA synthesis without directly binding to ribosomes or cell wall structures. It instead inhibits key enzymes involved in bacterial metabolism, which overall disrupt cell energy production and inhibit growth. It reduces inflammation mainly by inhibiting reactive oxygen species in immune cells, a benefit for inflammatory skin conditions like rosacea and some types of acne. Figure 1 is from an article published on Semantic Scholar. I like it because it clearly illustrates 5 distinct pathways ACA uses to help reduce inflammation. With part 1 showing blocking of a neutrophil activity, you may wonder, why would you want to block a white blood cell's activity? Well, because neutrophils can contribute to excessive inflammation and tissue damage. While they are essential for fighting infections, they release reactive oxygen species, denoted as ROS, and pro-inflammatory signals that can harm healthy skin cells if overreactive. Through part 2, we can see how ACA indirectly inhibits toll-like receptor 2 activation, which is responsible for recognizing bacteria and triggering inflammatory responses. This reduces the immune system's overreaction to bacteria on the skin. Additionally, CD36 plays a role in amplifying inflammatory responses, and we can see that it is also blocked by AZA in the figure. CD36 is a receptor involved in recognizing bacterial components and triggering immune signals, including the production of reactive oxygen species. Excessive activation of this receptor can lead to chronic inflammation and worsen skin conditions like acne or rosacea. Part 3 shows the NFKB pathway, which AZA blocks the activation of. NFKB is a transcription factor that controls the expression of pro-inflammatory cytokines. This reduces inflammation at the genetic level. Through part 4, we see peroxisome proliferator activated receptor gamma, which is a receptor that reduces inflammation and promotes skin repair. That we want more of, and that is what we see occurring here in the figure through the use of AZA. Additionally, more of that is helping to further block the NFKB transcription factor, which is what we don't want much of if our goal is to achieve healthy skin. Lastly, part 5 shows the blocking of kallikrein 5, which is an enzyme involved in skin inflammation. On the far right, we also have cathelicidins, antimicrobial peptides that help the skin fight infections. Now let's take a look at a figure that shows the anti-keratinizing properties of ACA. In general, AZA decreases keratin buildup, preventing clogged pores and reducing acne formation. Figure 2, part 1 illustrates how AZA inhibits certain molecules in the mitochondrial respiratory chain, leading to reduced ATP synthesis. This decrease affects ATP-dependent processes, including the fibroblast growth factor receptor signaling pathway, which is important for functions like cell growth, but upregulation of this FGFR2b has been linked to acne-prone skin. In part 2, we see how AZA reversibly inhibits DNA synthesis and cell proliferation in keratinocytes. Lastly, part 3 shows the reduction of filigrin expression in these keratinocytes. Filigrin is a protein involved in skin structure and barrier function. However, this protein tends to be overexpressed in many skin conditions, so that's why regulation of expression of this protein is important. ACA selectively targets hyperactive melanocytes, inhibiting tyrosinase, an enzyme involved in melanin production, which may lead to hyperpigmentation reduction. And lastly, but most importantly, the low risk of bacterial resistance. Since AZA doesn't directly attack bacterial cell walls or synthesis machinery, it does pose a lower risk of resistance development. All these attributes make AZA a versatile treatment option for acne, rosacea, and hyperpigmentation without the typical concerns of antibiotic resistance. It's important to note that since AZA is a saturated dicarboxylic acid, like we said earlier, it's poorly soluble and requires a good solvent to penetrate the skin effectively. It is generally gentler than many other cosmeceutical products, though it may not be suitable for all skin types. It may cause some mild skin irritation. And finally, there's the issue of selective toxicity. This raises the question, is the concentration required to eliminate harmful cells safe enough to avoid damaging healthy skin cells? And if so, AZA could be considered safe for long-term use. In my research, I aim to determine if the antibacterial properties of ACA could effectively target the escape pathogen panel relatives. To test this, we grew escape relative bacteria by inoculating broth cultures from stock slants and incubating them for 48 hours at 37 degrees Celsius, as shown in figure A and B. After visible growth, we used each culture to create a bacterial lawn on each agar plate, shown in figure D. Next, we took some time to prepare various AZA concentrations, 5%, 10%, and 20% in dimethyl sulfoxide, and soak sterile filter disc in these solutions, carefully placing one of each concentration on the agar plate with each bacterial species. Control disc included DMSO as the negative control and standard antibiotics, penicillin, a narrow spectrum antibiotic, and tetracycline, a broad spectrum antibiotic. Plates were then inverted and incubated for 24 hours at 37 degrees Celsius, and growth inhibition was measured in millimeters. In the next slide, we will take a look at the zones of inhibition of two of the species tested. Here are the results of the zone of inhibition assays for K. urogenes shown at the top and B. subtilis shown at the bottom. The control DMSO labeled as C showed no inhibition, while tetracycline labeled as TE and penicillin labeled as P showed expected substantial inhibition. Lastly, AZA treatment showed concentration-dependent inhibition increasing from 5% to 20%. Statistical significance was determined by ANOVA with Dunnett's post hoc analysis, and ANOVA being an analysis of variance used to compare the means of 3 or more groups to see if there are significant differences between them. It tells us if at least one group is different, but it doesn't specify which one, which is why we follow up with Dunnett's post hoc analysis to see if the result is actually significant. This test compares each group to a specific control group to see which groups differ from that control. The graph shows inhibitory effects of ACA, 5% to 20%, on multiple escape relatives, B. subtilis, K. urogenes, S. epidermidis, and E. coli. DMSO, like we said, served as the negative control, while penicillin and tetracycline were the positive controls. The error bars here indicate standard deviation, while the subscripts show the different levels of significance. In the next slide, we'll talk a little more about what these results mean. As we said previously, DMSO showed no inhibition of bacterial growth, whereas all 4 escape species tested showed significant inhibition at 20% ACA, with inhibition increasing proportionally across the ACA gradient. Additionally, tetracycline and penicillin significantly inhibited growth in all species except S. epidermidis, which, interestingly enough, 20% ACA had the most significant growth inhibition in S. epidermidis, a common skin inhabitant, compared to other treatments, compared to the 2 antibiotics. So I've reviewed several studies that explore different formulation vehicles to enhance skin permeation of ACA. However, I have not yet come across one that includes evening primrose oil, which is derived from the seeds of a plant native to North and South America. So I would like to explore this farther in future research. I'd like to take a moment now to thank the Yarborough Grant for funding several resources used in this project. I'm also grateful to Dr. Rushing for providing us with escape panel cultures and to the Catawba College Biology Department for supplying additional materials. Lastly, thank you all for taking the time to tune in.